Welcome to the second installment of Biolyfe webinars with myself and Dr. Peter Schulz. We're going to start calling these webinars Biolyfe live learning sessions. We want them to become more of a discussion, so they're less of a lecture, and just to make it more consumable for everybody. So instead, I'm going to ask him some questions about today's topic. Today we're going to address epigenetics. This might sound scientific to the average person, but that's what this is all about. Simplifying the science, and most importantly, taking back control of our biology. So we're obviously dealing with genes here. We know that genes are segments of DNA that are encoded with instructions for building and maintaining living organisms. The traits that are passed from parents to offspring. Now, epigenetics is the study of environmental factors that can influence the genetic code. Doc, could you elaborate a bit on that and give us an understanding of where science and society are with epigenetics right now?
Yeah, absolutely. Thanks, Brian. This is a really exciting field. I've been interested in this for a number of years and studying it pretty intensely. And it's really an emerging thing where previously we had kind of been, we've been told that our genes are health outcome. This is what our fate is. You know, back a number of years ago, a long time ago, Watson and Crick, they discovered the DNA double helix molecule and said, okay, well, this is pretty interesting. And then along came the Human Genome Project in the 90s. And we kind of thought, all right, cool. If we can figure out all of the genes in our body and what they do, then we know what's going on. So that took about 10 years, and this is kind of what we've been riding for a long time. But it's what's not true is that our genes don't actually code for something really specific. The way it actually works is you have, initially they thought, okay, there are over 100,000 proteins that our body makes. And the genes code for proteins, right? And they thought, well, there must be over 100,000 genes, 140,000 genes or something. Well, it turns out that there are only 20-something thousand genes. And that's not very many genes, we're not far off from a simple worm. It's almost the same number of genes. The difference is we can produce over 3,000 different proteins off of one gene. And that's how we determine what is going to be built and what's going to be made. So the way that actually works is literally epigenetics. It means above the genome, above genetics, but it is determining what parts of the DNA get read and what proteins get produced off of that. And that's actually what really ends up determining our health outcome and what's going on in our life, outside of genetic disorders, of course. This is how we get from a fertilized egg stem cell all the way to a human being.
That's interesting. Another thing that's often spoken about at the moment is cell differentiation. When we talk about stem cells, we think about the foundational building blocks of tissues and organs. What we don't think about is how these stem cells are differentiated. Could you explain that a little bit? Is it all inherited genes, or are the factors that change that are there factors that change that coding? Does the fact that my father had prostate cancer give me a higher chance of the same fate?
That's a really good question. So our genes are more, they correlate with certain health outcomes more than cause outcomes. So when you have a familial history of something, my father, my grandfather, everybody had prostate cancer or had heart disease or whatever, it doesn't mean your genes are coding for that. It means that there is a higher chance that those genes can get switched on. When you talk about cell differentiation, this is a fascinating topic. So we start from a fertilized egg, right? It's one cell. This is called a pluripotent stem cell, and that is half DNA of the mother, half DNA of the father. And then that cell divides, and we have two, and those cells divide, and we have multiple of these pluripotent stem cells. Well, that still doesn't do anything yet. Those have to start down a pathway of differentiating. We need some of these stem cells to become neurologic stem cells. We need some of them to become connective tissue stem cells, other organ stem cells, and that has to be programmed. So the neat thing is every single one of these cells in our body, our skin cell or brain cell or muscle cell or even these stem cells, they have your whole genome. They have every gene inside of them. And the amniotic fluid that surrounds the baby, that has the programming that tells these cells, okay, now we're going to build a liver. And so what happens is all the genes that are inside that cell, it turns on the liver genes. It keeps all of the other ones silenced and kind of covered up. It turns on all the liver genes, and now all these cells become liver cells. They have every one of your genes in there, but now they're just a liver cell. This happens then throughout life as well. It gets altered, it gets changed, and that's a lot based on what we do. And we can influence those things. So that's literally what epigenetics is, and how cells start from a pluripotent stem cell and then become differentiated into different cell lines. And this is all done by the amniotic fluid.
Great. Another thing that's been asked about a lot, and what I've seen online a lot, is biological age.
Mm-hmm.
It's a term that's been thrown out there. What is the difference between biological age and the amount of candles on my birthday cake?
Yeah. So, how old you are, how many years you've lived, doesn't really correlate with how healthy, how fit, how strong, how mentally sharp, how good you feel or how free from disease you are. Biological age does. These types of biological age clocks, you call them. The most accurate ones are DNA methylation clocks, and that is literally epigenetics. We're measuring the gene expression in your adult body. That is measuring how well your genes are currently expressed. And biological age is what determines basically your risk of disease. And we're talking about all-cause mortality, and then especially all the major diseases that people end up with and that are chronic diseases. Your risk of those is lower, a ton lower, when you're young, when you're 20 years old. You simply don't have very many 20-year-olds get cancer, and you have more 75-year-olds that get cancer. This is literally because of their biological age. If we took a 75-year-old and measured their biological age and it said that they were 56, or we did the different things to reduce their biological age, their risk of having cancer, for instance, is that much lower. And when we measure that stuff, it's all epigenetics. It's all DNA methylation. So the way this works is we have, this might get a little complicated, but it's upstream regulation of gene expression. So we have chemical caps that sit on the genes basically and protect it from being read. Along comes RNA and tries to read that, but it's protected so it doesn't read that. When I talked about cell differentiation, if you've got a skin cell, all the skin genes are on, you've removed those chemical caps, RNA reads that, produces the skin proteins. As we age biologically, and this is through life, through damage and lack of repair and so on and so forth, we end up with genes that are incorrectly expressed in the same cell. Take a skin cell, for instance, and this is a really good example because it's visible. And you have that skin cell and it gets damaged, say by UV light. And now the proteins that are getting produced off of the DNA in that cell, it's incorrect. And so you've removed some of these methyl caps off of other genes. Improper genes are being expressed. It still kind of looks like a skin cell, kind of behaves like a skin cell, but it isn't, and it starts to divide. And what do we call that on our skin? A rash. These are a bunch of cells that kind of are confused, don't know what they're supposed to do, and they're not functioning very well. And that's how it relates to your health outcomes. And that's exactly what we're measuring when we're measuring biological age. It's the DNA methylation, it's epigenetics in your whole body, and we're doing that on a system-by-system basis so we can identify which systems are keeping up with regeneration and which ones are failing to keep up.
And how is that tested?
So it's this simple, either a finger prick,
Okay.
and we get a drop of blood out of your finger, or it depends if we're doing some other things. We'll draw a couple tubes of blood. We just need a couple drops of blood really for that. Yeah.
So knowing that we can lower our biological age,
Mhm.
and that will result in a longer, healthier life, what, in your opinion, are the three most important parts of our lives that we can change in order to lower that biological age?
Yeah. So really, there are main factors that we can all do. Every single one of us can do it, and the consistency of that throughout our life is what establishes real health. You can't do something for a couple of weeks and expect it to have that big of an impact in our bodies. And there are really four main things that I would point to. Nutrition. And there are a lot of details about nutrition, but mostly the most important things are that we're eating clean, natural, whole foods that are unadulterated, unaltered, unpreserved, processed, and that kind of stuff. Then you have sleep. This is when we regenerate and repair. We need different types and levels of sleep, and it needs to be consistent. We need to focus on making that sleep-wake cycle, the circadian rhythm, really good. Stress resolution. That's super important. We have to do that every day. And then exercise, movement and exercise. I mean, if all the other things, stress resolution, sleep, and nutrition, and maybe supplements or medications all combined to equal 50% of your health outcome, exercise is a full other 50%. Movement and exercise, if we've got to keep moving. And that's one of the main things that we are focused on here, is that if you have an injury or orthopedic problem or pain that prevents you from doing your normal movement that you would do in a day, then you have to fix that stuff. You have to get that better so that you can keep moving.
Exercise.
Exercise, yeah.
I've done some pretty extensive research on overall health and longevity programs. Your system is slightly different. Could you touch on how Biolife's membership packages and why you design them that way?
Yeah. This is what gets really exciting, I'll tell you what. By doing these healthy lifestyle things, the research shows that basically you can add a healthy 12 years to your life compared to somebody who doesn't really do that many of them. So the difference is the 70-year-old who can do everything they want to do, they might be wanting to work or travel or physically do everything and be mentally sharp, and compared to the 70-year-old who is in and out of hospitals, has a lot of disease and having a lot of health problems and basically degenerating pretty quickly. The difference there can absolutely be done by those healthy lifestyle things that I was just referencing. And that's nice to be able to add 12 years to your life, 12 healthy years to your life. Now we should do that, and we'll help guide that. But what we really are doing, and what these programs are, and people who are doing these programs, what's happening with them is that we're not just adding 12 healthy years to your life. People are being able to look at this and measure and see that they're actually reducing their biological age. Now they're confident that their risk of disease, these chronic diseases that end up taking people down, are so far away that they have confidence that they can keep going as long as they really want, right? I mean, we'll see how it all goes in 20, 30 years, but overall, you're talking about blowing that 12 healthy years out of the water and saying, well, this all can work for a lot longer. And so each one of the aspects of our memberships or the programs that are established here at Biolyfe are attacking epigenetics and other hallmarks of aging at different angles. So we'll be touching on a lot of hallmarks of aging, and epigenetics is one of the big ones. And what we're going to mostly be talking about here is that things that you can measure and test, and then things that you can do something about in those hallmarks of aging. So epigenetics is huge when we do that, and that's one of the aspects that we're attacking or addressing when we do different therapies that we've designed in these programs. Some of the other aspects that we'll touch on later, some of the parts of the programs will be specific for that. But that's why we've set them up the way we have, to be able to address each one of these hallmarks of aging that we know we can measure and do something about, and do something about them to literally lower our biological age. And as research continues, and it's moving fast these days, we're keeping up with that research, and we'll move along with it and say, well, look, now we can address this part of aging and this part of health. And it's not replacing health. It's not replacing a mechanism with your body. It's making your body do it again. That's what our whole aim is.
I think that's as easy as it's going to get for epigenetics. So thank you very much, and a lot of the information and a link to Biolife's membership packages will be in the description below. If you want any further information or have any further questions, please feel free to get in touch with us. You can get hold of the office or contact us online. I hope it's been an informative one, and you're one step closer to taking back control of your biology. Look out for our next Biolyfe live learning session. Thanks. Goodbye.